we voted on topics, and the one that had the most votes was the genetics of mass killers. And so that's what we're gonna talk about, the genetics of violence. So to get ready for that, I have a series of articles. These are easy to read because they're like New York Times articles or other kind of popular media articles, rather than like a science article. But I have a primer assignment I want you to do before coming to class. Is so just a single page, and I'll show you kind of what's on that. It has the links on this assignment. So there's these 3 articles that you need to read, and then there's just some terms that you can define. And these are not terms that are necessarily directly in there, but these terms will kind of come into play when we discuss, the genetics of any complex trait, like a trait like violence. And then I have, of course, a major part of this discussion will be about nature versus nurture. So this is kind of in your own words. You can kind of say what you understand about nature versus nurture. And then the last part of it is looking at what are some genes that have been implicated. They have found some genes that have been in families where there's like many violent offenders. They've tracked down and looked at genes that are mutated, and so they have a couple of kind of candidate genes that could be linked to aggressive behavior. But of course we'll talk about how does that then determine the trait. So the links are there. They're also in the module here. So just make sure that you do that before coming to class. Look at those articles. Okay, does anyone have questions about that? Yeah.
So is this?
This is different. That's for our genetics, like the lecture stuff.
Oh, okay.
So the discussion is more kind of just applying what we're learning with genetics to a kind of real world scenario. Don't worry about, I don't think you should be able to see any of the PGD, but if you saw a PGD primer or anything like that, we're not going to do that one. So instead we're doing this other topic. All right, and then there's a video here. We're gonna watch that in class, but don't worry about watching it ahead of time. Okay, any questions or anything about that or the end of the quarter? I know we're down to, we can count on 2 hands, 7 days left of class, right? Anybody have any questions about the exam, final exam, or the final exam is not cumulative, it's only the most recent stuff that
Will this genetic discussion, the serial killer gene thing, will it be in the quiz or the exams?
No, this stuff is, genetics discussion is kind of a side topic. It's not covered, the content is not covered on any quizzes or exams. And also, since we've had the biotech stuff was on the last exam, it's not covered on the final exam. Okay. All right, well, I thought we could actually start with just some review questions since we kind of got through the bulk of the meiosis. So if everybody wants to get out, you can have a phone or a laptop. You can go to socrative.com and then do student login, and then the room code is Miller Science. Sounds like a beer. I have to change that now. Think of something creative. And we'll just kind of see if there's any areas that we should review before we go on to actually talk about genetics. Okay, so the first question says, during meiosis, the sister chromatids separate during which of these phases? All right, good. So almost all got it correct. So the one we're deciding between is the 2 anaphases, because that is the phase when the chromosomes come apart, and then you just have to decide when are the sisters actually separating. So in anaphase 1, what is it that separates? What comes apart at anaphase 1? Yeah. Yeah, the homologous chromosomes are coming apart. And then the sisters will come apart in anaphase 2. So yesterday we briefly talked about how is it that the sisters stay together. Does anybody remember the name of the protein? Shigoshin, right? So that's guardian spirit in Japanese. And so that's the thing that basically recruits a phosphatase, gets rid of the phosphorylation around the sister chromatids during anaphase 1. And that allows them to stay together so they don't get cleaved by separase. Okay. So, genetic recombination takes place during which phase? Okay. All right, so yes, prophase 1. So the only, when we look at mitosis, meiosis 1, meiosis 2, which broad phase, or which process, I guess, does genetic recombination take place in? Yeah, meiosis 1. And then specifically within meiosis 1, it's in prophase that we have synapsis, where the homologous chromosomes come together, and then there is that physical exchange that happens, and that only happens in prophase 1. By prophase 2, why is it that genetic recombination cannot take place? What's already happened at that point?
The homologous chromosomes are separated. So
Yeah, exactly. So those two are no longer even in the same cell, so this is only happens in prophase 1. Okay. So when does ploidy change? Which process? Okay. We'll take a look. Almost verbatim. All right, so meiosis 1 is the only time it changes. If we look at mitosis, it's 2N, stays 2N. Meiosis 1 is the only phase where the diploid, 2N, goes to haploid. So it goes 2N to 1N. And then in meiosis 2, it stays 1N. It's 1N in the beginning, it's 1N in the end, and the only thing that is changing is the sister chromatids are separating. Okay. Chromosomes decondense during which phase? Okay. See? Okay, good. So most people got it right. So telophase is when the chromosomes are decondensing. What else is happening in telophase along with that?
nuclear envelopes reform.
Yep, the nuclear envelope is reforming. Yeah, the spindle's gonna be breaking down and everything is, this also happens kind of alongside of cytokinesis, so that would also be happening usually around the same time. Okay. Bivalents or tetrads line up at the metaphase plate during which phase? Alright, good, so metaphase one is the only time that you would have giblets lined up. The other phases would just have individual chromosomes lining up. So one thing I often do on a quiz or an exam is to draw a picture, I myself draw it. But, it'll be of one of the phases, and usually I give you information, like, I might say, 2n is equal to 6. And this information is important because it tells you how many chromosomes there should be, which will allow you to figure out, is this a mitotic cell, or is it in meiosis 1, meiosis 2? So using this information, if 2n is equal to 6, that means that in a diploid cell, there should be 6 chromosomes, right? If you're looking at that cell, and let's say that I have 3, and maybe also I have some crossover events that have happened here, you know, or maybe this one is actually green. If you saw a cell like this, you know right away that I have 3 chromosomes, so which phase would that have to be in? Is it in mitosis, meiosis 1, or meiosis 2? It has to be meiosis 2 because the ploidy number has already been reduced down to haploid. And then which phase would you say that looks like? Yeah, so this would be metaphase 2. If you saw, instead of this, if you saw, you know, 6 of them lined up randomly along this, then you would know, all right, there's 6 chromosomes here, they're lined up, and I see that I don't have tetrads, so then what phase would that one be? Yeah, metaphase of mitosis, right? Because I know that they're individual file. So you have to kind of use the clues of the numbers of chromosomes to figure out what phase they're in. Oh, we're gonna skip this one, actually. So this one I took out of okay, forget that one. We're not gonna do that one for now. Okay, so that's kind of the basic review for meiosis. We just have the last couple of slides. How do I unplug this? I can't see my Dropbox whenever I have it plugged in. Okay, so let me open up the meiosis lecture. And we just had the very end of it left. So we talked about differences. So we kind of left off of this. We haven't yet talked about, I guess we kind of mentioned it, but with meiosis, the goal here is to create these very genetically variable gametes. And so there's different mechanisms that help to generate that genetic diversity. 2 of the mechanisms happen during meiosis, so in the actual creation of the gametes. Can anybody remember what is generating the variability between those cells? What's one thing? Crossing over. Yeah, so crossing over is creating combinations that have never been seen before. That's one thing.
In the lining stuff, they kind of just do it randomly.
Yes, so you could say random alignment, or also there's another word you could say independent assortment of the chromosomes. Those both describe the same thing. So the first one I have listed, homologous recombination, is the same as crossing over. So that increases the number of possible allele combinations. To answer that first question, what specific stage does that happen in? Again, you kind of already said it, but prophase 1. So in your notes, you can add prophase 1 to when this happens. Independent assortment happens because as those homologous chromosomes line up, they can line up with maternal on one side and paternal on the other, or they can swap. And every single chromosome gets to independently line up randomly. So when would we say that happened? Metaphase 1, right. So those 2 happen during meiosis. The third factor that generates genetic diversity is actually the fertilization itself. The fact that we have random fertilization of one gamete one sperm with one egg, and that increases the diversity of offspring that are possible. So just back of the envelope calculation to figure this out, how many possible combinations we could create, and this doesn't even factor in crossing over, so it's an underestimate. But if you say the independent assortment, there's 2 to the 23rd possible arrangements. That means that there are 8.4 million possible gametes on both sides. We multiply those together because of fertilization. All of the possible combinations we could make, and that's 70 trillion possibilities. So all of you are one of a kind. So I have I don't know, have you guys ever heard the Dr. Seuss poem, what is it, like, you are the only you are, anyway, I think I put that poem on your test, so. And the question is about what factors can create this genetic diversity. Okay, anybody have any questions on that? That makes sense. All right. This picture it helps to describe that random alignment or the independent assortment idea, which is that you've got different ways that these chromosomes can line up, and that's going to create different combinations of different genes. And this is, of course, only when genes are on separate chromosomes. Okay. We will come back to this too in genetics, because this is how when we do the dihybrid cross and we have a Punnett square, that's how we figure out what are all the possible gametes you could get. Okay, so we've already done that. The very last part is about gametogenesis. So in the context, we've kind of gone over it, just what are the basic steps of the process, but in the context of human gametogenesis, there are some specific details that I like to point out so you understand how these things happen, and I'm sure you've learned about it before. But for creation of sperm, spermatogenesis is the name of the process. It starts with the spermatogonium germ cell, which you can see starts out as a diploid cell. That cell undergoes an asymmetric division in that it creates another spermatogonium cell that is going to maintain that population. The other cell is going to go down a path of meiosis. So it's going to actually divide several times to go through the process of meiosis. That cell is called the primary spermatocyte. Again, you can see that it starts as a diploid cell. After meiosis 1, we have secondary spermatocyte, which is now haploid. So that was our first division. This division takes, these 3 different divisions take about 70-ish days to happen to complete the whole process. So for males, after puberty, they're going to be creating sperm their entire life, which is why you read in the news there could be a 74-year-old father of a child, right? They're still creating sperm. And the process is happening continuously throughout the lifetime. The secondary spermatocyte will then undergo meiosis 2. That creates spermatids, which is just actually the content of the genetic contents there. In order to create a sperm, there's other processes that have to happen for that maturation of the sperm. It develops a flagellum. It also has several things that are in the head of the sperm, which are used for fertilization and for species-specific fertilization. So there has to be some interactions between this sperm so that it can only fertilize an egg of the same species. And there's also things in here that help to break down the outer layer of the egg. So there's a lot of enzymes and things that are also part of this maturation process. Okay. So that process in males pretty straightforward in that it's happening continuously. All of the 4 spermatids can create mature sperm that could fertilize. Females have a slightly different process. And this is true of female mammals, and specifically we're going to talk about what happens in humans, what happens for oogenesis, or the production of oocytes. So again, we start with the germ cell. This is called an ovonium for females. The first division, just like in males, is unequal in that one of the cells maintains the population. The other cell is going to undergo meiosis, so that's called the primary oocyte. This undergoes meiosis 1 to create the secondary oocyte. Now, one thing that's not so clear in this picture, which should be shown a little bit more clear, one of these cells is not going to become a viable egg. It will become instead a polar body. And the polar body actually, it should show unequal division of the cytoplasm because the polar body is much smaller than the oocyte. Okay, so why, can anybody kind of make an educated guess as to why there is unequal division of cytoplasm for the female side, but not for the male side?
I feel like it's because the egg is kind of taking on the responsibility of doing all the work. And the sperm
Yeah.
is kind of adds extra DNA.
Exactly. So if you look at the size of the egg versus the size of the sperm, and we saw that picture of an egg with all those sperm, the sperm was really are so small in comparison to the egg. So the maternal side, the mother, provides all of the cytoplasm, most of the organelles, most of the proteins, really stockpiles everything for the zygote. So because of that, when it is when it's undergoing meiosis, one of the cells gets most of it, most of the cytoplasm. The other one becomes a polar body. And to give you an idea of the difference here, this is a polar body. So the size of a polar body is much, much smaller. That should be a lot smaller of a picture. The second part of meiosis only will happen if fertilization occurs. And fertilization occurs in the fallopian tube. If that happens, then the egg cell, or the oocyte, I should say, will complete meiosis too. If it never gets fertilized, it gets sloughed off, and it never completes meiosis. The other really interesting thing about females is that there is a pause. The primary oocytes. So let's actually talk about when this happens. For all of us females in the room, we created all of our primary oocytes when we were fetuses. In our mother's body, we had all of the primary oocytes that we will ever have in our lifetime. They all arrest in prophase 1. So specifically, they arrest at the stage when they're crossing over. And they're just stuck there. And think about how long they stay in that phase, right? If you're a fetus, you don't resume meiosis until puberty. And then it's only a few of your eggs will resume, or a few of your oocytes resume this process each cycle. And so really, if you're like 40 years old, like myself, this has been how many years? 40 years where your oocytes have been stuck in a phase, right? And so as you know, as women age, they become higher risk for chromosomal abnormalities and things like that. And that's because your oocytes have been in dusty, getting freezer burn in your ovaries for all those years, right? So make a note in your notes that oocytes are arrested in prophase 1 until puberty, and then each month, a couple of them will begin this process of maturing. They only get to metaphase, and then if there's no fertilization, they get sloughed off, and they never complete meiosis. So there's 2 pause points, prophase 1 and then metaphase 2. They'll pause. The other interesting thing, this is something that for females to kind of think about, since you have all of your oocytes when you are a fetus in your mother's body, that means that you actually at one point were a primary oocyte in your mother when she was a fetus and her mother. So there's 2 generations that were actually together in the same place. I just find that really interesting to think about that. Like at one point, you were in your grandmother when your mother was a fetus. So anyway, that's just kind of interesting. I guess the other part that we should talk about is after this divides here, you create a polar body. The second division also creates another polar body. So there's actually only 2 polar bodies. I don't think this one divides. I don't know, this textbook shows that this divides and you create a polar body, but I don't think it continues. This, you actually create 2 polar bodies and 1 egg, what becomes the ovum, the egg. So when you can actually usually see those polar bodies actually attached, like when we look at oogenesis and other organisms, you can see that there's 2 polar bodies that have been released, and these just get absorbed by the body. Does anyone have questions about either of the processes? And I guess the other thing we can talk about for females, do you produce eggs or release eggs your whole life? Or is there a point at which no?
They, around 50 to 60 years old, you start reaching ovarian failure and you run out of eggs.
Yeah. So you, after menopause, then women do no longer release eggs. So there is a time limit during which you are producing eggs. And as I said, with age, generally there's more errors in meiosis. And so I have a couple slides about that as well, as to what will happen usually. So the major problem with meiosis, when meiosis, like we're saying with older women, one of the things that can happen is non-disjunction. So non-disjunction is an error that happens when you don't get complete separation. It can happen actually at multiple different time points. It can happen in meiosis 1. If non-disjunction happens in meiosis 1, you see that the homologous pair gets pulled to one side. That's going to result in aneuploid cells that have the wrong number of chromosomes in the set. It can also happen in meiosis 2. So sometimes it doesn't happen at meiosis 1, but instead happens at meiosis 2. But in either case, you get aneuploid cells, some that have extra chromosomes, some that are missing chromosomes. So the blank here should be results in aneuploid, this word right here. This causes those chromosomal abnormalities that we've discussed. We mentioned Down syndrome. So that one is where a person has an extra chromosome 21. There's also cases where the sex chromosomes have non-disjunction. So I was going to share with you a couple of other, the Turner syndrome and Klinefelter syndrome are both caused by sex chromosome non-disjunction. So to look at these, and when you test for this, one of the things you can use is a karyotype, where they basically collect all of the chromosomes from an individual. When they do this, they don't appear like this naturally. You actually have to use computer programs to align and figure out which ones are chromosome 1, 2, etc. They do that based on size and also based on the banding pattern that they have. So the computer would then generate an image like this, and you can compare all of the chromosomes and see if you have the right number of copies of each chromosome. If there's any chromosomal translocation, sometimes you can visibly see that. You can also, nowadays they don't just do this test, but they also look at genetic markers. So how many copies of a specific marker do you have? And that can be even more sensitive than just using a visual like this. So this would be a typical karyotype where the individual has 2 of every single chromosome. In this, is this a male or a female that we're looking at? Female. You have 2 X chromosomes, so we know this individual is a female. If we look at some others, in this case, this is another example of karyotype. So can you identify the disorder based on the chromosome array here?
Trisomy 21.
Yeah, so we have 3 21 chromosome 21s. And is this male or female for this one? Yeah, so we have a male with Down syndrome. So Down syndrome is most commonly caused by chromosomal non-disjunction. Usually it's coming from the side of the mother, because as we said, usually the age of the mother can increase the likelihood of having these types of non-disjunction events. This one here is another example of a chromosomal abnormality that is tolerated. Remember I said that you can have other cases where there's trisomies, but usually they're not viable and the body will spontaneously abort the embryo. These ones with sex chromosomes are tolerated. So XXY is another abnormality that causes Klinefelter. And that would mean that that person had an extra chromosome. So it's called 47XXY. This one, because you have 2 X's, we'll talk about sex determination a little bit in this class, but not in a huge amount of detail. The Y chromosome is actually what determines the male basically the male sex characteristics. So if a person has Y and they have this region of the Y chromosome that's intact, they will develop testes. It sends them down the track to basically develop into males. But in this case, because they have an extra X, they also have some secondary sex characteristics. So in this case, they have breast tissue, also low testosterone levels. And in this case as well, these individuals are infertile. They cannot produce gametes themselves. The other one that is pretty common is that X, it's called XO, where you just have one X and you don't have a second sex chromosome. So given what I just told you about how sex is determined, if you had XO, what would you think primarily the characteristics would be for that individual, male or female? Female. So they primarily have female sex characteristics, but there are also issues of infertility, and there's intellectual progression issues, learning disabilities, and things with that as well. And that probably is because there's a lot of genes found on the X chromosome, so it does a lot of different things. Okay, so we talked about these errors. Why are they more common with age? This is just a graph, just looking at Down syndrome, but the incidence as age increases. So you can see, early 30s, it's starting to increase, going from 1 in 1,000 to 1 in 300. And then very quickly it jumps up, like 1 in 100 by 40. And then it kind of skyrockets after you're 40. So even when you're 35, because I had my kids when I was like 30 and 32, but they already start to treat you like you're like a geriatric. They actually call it geriatric pregnancy when you're like 35, which is very like, wow, I don't feel that old. Or AMA is advanced maternal age at age of 35. And then they would recommend more testing for you, just so that, if you have a chromosomal abnormality, some of them, like Down syndrome, are viable, but others are not going to be viable. And so sometimes that's, a matter of, do you want to carry a pregnancy if you know that you have, say, trisomy 13, which is your child will not make it, and do you want to go through the trauma of that? So I have a couple of friends that went into the genetic counseling route, and one of them does prenatal counseling, and so that's the decision that's what they usually counsel about is, if you're testing and then you find something, you have to decide, do I want to keep this or do I want to go through that? So for the blanks for this chart, it says eggs are arrested in prophase 1 until puberty. So that would be or ovulation, I guess, could go in the blank. So every month when you begin ovulating, a few of the oocytes will begin the process of maturing, but only a few every time you have a cycle. And then they arrest a second time in metaphase 2 until fertilization. So the second blank should be fertilization. Okay, so that's all the slides I have for meiosis. Does anybody have any questions? Okay. So now we get to genetics, which personally was one of my favorite topics in biology. So a lot of this I think you do go over in 160. So we'll talk about sort of the classic Mendelian genetics, which is the monohybrid, dihybrid crosses, Punnett squares. I know that everybody probably knows how to do those. And then we'll talk about some things that you may or may not have talked about in depth, which is some of the variations, some of the interesting things with different dominance patterns other than just complete dominance. Gene interactions, so epistasis is a phenomenon that happens when genes can affect whether or not the others are expressed. And then things like incomplete penetrance and expressivity. These are kind of the interactions part that makes genetics really complicated. The last part will fit in really nicely with our discussion tomorrow about violence, because these are complex traits that are affected by more than one gene, but also affected by environment. So as you know, there's a lot of traits, What are some others that you can think of that would be considered complex, that are not just determined by your genetics, but also have environmental inputs? Anybody think of some traits that are not simple traits? Yeah.
Cancer.
Cancer, yeah, cancer does have a lot of inputs. It's multiple genes and also environmental factors. Yeah.
Alzheimer's?
Yeah. So yeah, that would be a trait, getting that, again, many different genes involved and also environmental.
Maybe something like your sense of humor.
Yeah, some complex personality traits.
Is it like mental disorders like schizophrenia or?
Yes, exactly. So those ones also have inputs from both genetic factors and it's not just one gene that's involved. Things like height, anything about height, intelligence, body weight, those are, as we know, they have some genetic inputs, but they also have environmental inputs, and people are really interested in understanding what's the balance of that, how much of your, traits are determined by the environment versus your genetic makeup. So we'll talk about how do they actually test that, and that's the idea of heritability, is trying to measure how much of a trait variation comes from genetics or the environment. Okay, so the reason why I love genetics is that personally it's just very interesting. Like, you can compare yourself to your siblings, trying to understand why you're similar to your relatives or different. Maybe you're really, really different from your relatives. These are my two kids. This was actually taken a long time ago, but you can see they look nothing like each other. They have the same parents and everything, but the oldest one is Jack. He has more of like my coloring, and then Charlie looks nothing like my child at all anyway. He's blonde hair, blue eyes, and looks very different. But obviously they are genetically related. Personality-wise, I think Charlie's more similar to me, and Jack is more similar to his dad. So it's just interesting. I think personal genetics is interesting to all of us. And then if you like animals, which I love dogs, it's interesting to understand how you get certain traits in dogs, even dispositions, and that's kind of the questions of how they set up breeding and what gets passed along. Other reasons for studying genetics, it has actually created all of the agricultural products that we use today. This is from thousands of years of basically selective breeding, right? Where people have said, I want to get the biggest apples. They breed things that have big apples, and over time, the apples become larger and sweeter, right? The ancestral plants look nothing like our current food that we eat today. This is actually teosinte, which is a grass that's in South America. That's the ancestral plant for corn. And just to show you the size of the kernels in the original plant, that's a quarter for comparison, so you can see how small those edible kernels were. After thousands of years now, of course, we have modern day corn, which we have further modified with genetic engineering, right? So kind of explains our food sources. It also helps us to track genetic illnesses. So kind of with the advent of understanding DNA biotechnology, people now have been tracing down for all of these different diseases, what are the genes that are involved? What are the genetic causes of the disease? Because if we understand that part, we can work on developing treatments. So there's really so many different applications for genetics. So most of the field of genetics was started. Does anybody know who we consider to be the father of genetics? Mendel, right? And he was a monk. What was his genetic model organism that he used? Yeah, so he happened upon the pea plant. Actually, it was fortunate that he chose that plant and that he chose the traits that he did, because it turns out that those traits were simple. They were inherited through simple patterns, and he was able to use math. He was actually a very well-rounded person. Like, he was a monk, but they sent him off to university because he seemed to be really smart. And he studied botany, physics, statistics, and he combined all of these to kind of make add, I guess, to the knowledge of how things were inherited. At the time, does anybody know how people thought traits were inherited? What were some of the ideas at the time? And this was like the 1800s. about traits? Did they even know about, at that time, did they know about chromosomes? Not really, right? So there wasn't an idea that discrete things were being inherited, and there was even ideas coming from like the Greeks that your traits were in your fluids, the humors of the body, that traits were passed along through that. At this time, there was a lot of ideas about blending inheritance, which it makes sense when you look at two parents and you look at their kids, usually they look like a blend between the two. But what Mendel discovered is that there was actually something discrete that was being handed off each generation, and it turned out that was the chromosome, and it was the unit of the gene that was encoding those traits. So his work, as we said, he used the pea plant. What are some of the reasons why pea plants would have been a good model organism? Especially after you've worked with worms this quarter, what are characteristics of a good genetic model organism?
They were inexpensive.
Can you say it louder? Inexpensive, yeah. Very cheap.
They have a short, growing time.
Yeah, so short generation time. Yep. Other things that are useful?
Easily observable traits.
Yeah, exactly. We talked about some of like, seed color or wrinkle versus round. Those were very easily characterized traits. Yeah. Oh, you
Cross fertilization and self-fertilization.
Yeah, so that's another really useful one that you've seen it with our worms. Like we only have to put a single worm and it will populate a plate. So that's kind of the idea behind self-fertilization. But also if you want to cross traits together, you could control the mating with the cross fertilization. All right, I think we hit on most of these. The only one that wasn't mentioned was large number of progeny, right? So with pea plants, all of those pea seeds are progeny, so that gives you large numbers. So why is it important to have large numbers with genetics? When you're trying to figure out how something is inherited, why is that useful? Yeah.
Larger sample size.
Yeah, so for statistics, the larger your sample size, the more meaningful your data is, right? So that's why if we were to do genetic experiments with cattle and they only have 1 to 2, progeny at a time, we're not going to be able to make conclusions about how things are being inherited very easily. So since that time, obviously we have a lot of other model organisms. We've talked already in our class about everything they did with the lactoperon, they did that in E. coli. We've kind of talked a little bit about Saccharomyces cerevisiae, yeast. They've done a lot of very basic science experiments with yeast. They've looked at like cell division and secretion because that's a simple eukaryote. Drosophila is very useful, and we'll kind of, in our genetics unit, we'll talk about how they used Drosophila. It's been used to study a lot of the sex-linked traits. And then obviously with C. elegans, we've already firsthand seen how we could use that to study something like Alzheimer's. And this I just shared. This is somebody that used to work down the hall from me at the MIH. This was at the International Worm Meeting. Did I show you this already? Yeah. So anyway, this just to show you like each of these communities has their own like international conferences, and they're a pretty tight group. Like people are usually pretty hardcore about their model organism that they study. So, all right. So coming back to genetics, a lot of this is review, so we're going to go through it kind of quickly, but just so that we're starting from the same page of definitions. What is a phenotype? So we're going to talk a lot about what is the phenotype of an individual and the genotype of an individual. Yeah, go ahead, Daphne.
A phenotype is the physical expression of a gene.
Yeah, so we could write physical expression of a gene, or an individual's observable traits, or physical expression of a gene. Physical expression of a gene. And then genotype, what would you say for genotype? How is it different from a phenotype?
Their genotype might not necessarily I guess, reflect completely what the phenotype is, because then I have like a recessive trait that's not shown.
Yes, so that, you are right about that. So for heterozygotes, you might not know that they're a heterozygote because they're expressing the dominant trait. So what should we say for a definition? For,
Specific sequence?
yeah, so it has to do with the sequence that they have. What did I write for? Yeah, I said the specific alleles or the specific versions of the genes that a person has. So specific alleles that an individual has. And we can't always tell that, like you were saying, that depending on what your genotype is, you may or may not know whether they carry a recessive allele, because we will typically only see the trait that coincides with the dominant allele. So for the example here, I have red hair as the trait, so that would be the phenotype. The genotype is determined by the gene that controls that trait, which in this case is the MC1R gene. And we talked about this same gene when we talked about pocket mice. Does everybody remember doing the pocket mouse activity? That is a gene that controls pigmentation. So even it controls fur color in mice, it also controls pigmentation in human hair and skin. So the MC1R gene, if you have the mutant variant of that, it produces a red colored pigment instead of the darker colored pigment. In order to have this red hair, because this is a recessive allele, you would have to have 2 copies of that same variant. So redheads have these 2 variant copies. All right, so we've kind of alluded to a lot of this, but because we are diploid, we have 2 copies of every single gene, and we can have the same versions of that gene on our 2 copies, or we can have different versions. The versions of the genes are called the alleles. So that is the definition for alleles, would be versions of a gene. With human nomenclature, we typically use a single letter, and usually the capital letter. Does anybody remember that actually? What does the capital letter mean versus the lowercase?
The dominant is
Yeah.
the capital letter, the lowercase is recessive.
Yeah, so especially when we only have 2 alleles, if we have a biallelic locus or gene, then we would have the capital letter is the dominant trait, and the lowercase letter is the recessive trait. Then we can describe people's genotypes depending on whether they have 2 of the same or 2 different alleles. So homozygous means they have 2 of the same allele. Heterozygous means they have 2 different alleles. So for these 3 genes, which are homozygous dominant? Which gene would be homozygous dominant? A, B, or C? Or A, B, or D? Everybody agree this one is homozygous dominant? Do we have a homozygous recessive? Yeah, this one, they're both little d. And then the B locus would be heterozygous. Okay, so we kind of covered that. All right, so kind of continuing on and talking more about dominance versus recessive. When we say something is dominant, how do we know that an allele is dominant? How can you tell if, let's say that you have a trait, let's say you have purple and white, and you have no idea which trait is dominant. Anybody know how you would figure out which trait was dominant trait? Or which allele was dominant? You wanna?
I think you can like cross the parents and then look at the progeny to discover what the phenotype is, and then you can figure out what the genotypes are in parents.
Yeah, so if you had a purple parent and a white parent and you crossed them together and all of the progeny were purple, then which one would be dominant?
The purple.
The purple. So basically when we try to figure out what is dominant, we look at the heterozygote. And that really defines which trait is dominant. So the dominant allele, this is my definition of it, the allele that produces the phenotype in both heterozygous and the homozygous form. So that means that whichever one you see in the heterozygous, that is the trait that is encoded by the dominant allele. The recessive allele you will only see when it has two copies of that allele, so when it will only be seen in the homozygous form. So kind of similar to what you mentioned here, if you didn't know, and let's say you have round and wrinkled, and you're trying to figure out which one is dominant, you cross them together. Because the parents are homozygous, they can only produce a heterozygous progeny, and then whatever trait that progeny shows will be which trait is the dominant one. Okay, so with this, we have round and wrinkled, right? Those are the two traits. I want you to think for a second, how could you explain this at a molecular level? How do we explain, this is the question here, what is the molecular basis for dominance versus recessiveness? And my hint is that genes code for proteins, so what is really the difference in the proteins that are encoded by dominant versus recessive allele? So why don't you talk, the person next to you, see if you can come up with an answer. How do you explain dominance versus recessiveness?
I think the proteins are more active. The proteins are more active. Yeah, that's what I heard. Anyways, and if the recessive is broken, I don't know. Broken proteins, so it has low expression. So it has low expression, so that the proteins are broken.
So it is encodes more.
For the dominant.
That seems in the same EV. Like the dominant ones are better at dividing, which is, I guess, good.
I watched the, what is it called? The lecture for the class, so. I try, but it doesn't really help.
All right, so what are you thinking about? How do you explain it at a molecular level, using kind of what we've been talking about this whole quarter, with what we know about protein function? Yeah.
It could be that the recessive is like a non-functional protein.
Yeah.
Or it could be that the dominant produces a protein that blocks the recessive.
Yeah, those are both good explanations, is that if it's recessive, maybe it's non-functional, so if you have a functioning copy, that's enough, and you see the wild type phenotype. Yeah, you wanna add on?
Well, I was also thinking maybe the promoter of the dominant gene is a lot more.
Yeah.
rare or something.
Yeah, so with dominance, that is a way that it can kind of produce something that's very hyperactive. So these are kind of the explanations that I have. If you have a dominant allele, you could have a promoter that makes more of that protein. So basically, you're kind of overriding what is produced from the other allele. It could be more active, so sometimes dominant genes are a hyperactive form, so that they're kind of like, regardless of what's being expressed from the other one, they're doing too much activity. Or the other times we see this is if a gene is being expressed in the wrong place at the wrong time. So irregular expression, it's not supposed to be there in the first place, so that it can show up no matter what, as long as you have, just that one copy. The recessive allele is usually loss of function, that something happened to that protein, it doesn't work as well, so it's broken, it could not be produced at all. Sometimes there's what we call a null allele, which means there's no functioning protein produced at all. So it could just be lower, or it could be completely gone. So if we have, think about our mutations that we talked about. We talked about nonsense, frame shifts. What type of allele do you think a nonsense protein would typically create? A recessive allele, most likely, right? Because you're going to create a protein that's not there, or maybe half of it is gone or something like that. That's probably going to make a broken protein. Which of these do you think is more easy if you randomly generate a mutation? Do you think it's easier to make a dominant mutation or a recessive mutation? So in other words, do you think it's more likely that we have loss of function or some gain of function? It's a lot easier to break things than it is to give them like superpowers, right? But it would take, let's say you have an enzyme, to make a dominant allele, you would have to have a very specific mutation to make it more active. But there's a lot of ways that you could break it, right? You could have mutations throughout. So for the most part, a lot of the mutations that we look at do create recessive alleles. There are, we'll talk about some examples of dominant, they do obviously exist. For the round versus wrinkled, here's the molecular explanation for this trait. Obviously, Mendel he didn't do molecular genetics. He didn't really have this understanding when he characterized this. But the R gene encodes an enzyme that's called starch branching enzyme, isoform 1, so SBE1. And the job of that enzyme is to convert the unbranched starch into branched starch. If that is mutated, so in the little r, that would create, if you had 2 copies of that, you would have this inactive enzyme. It does not branch. What ultimately happens initially, the peas swell up, but then they lose that water, and that's why they look wrinkled. So this is sort of understanding actually what is the gene encoded by that allele, what does that gene do? And with most of like human genetics, the goal is always figuring out for a trait, we have like a disease or a trait that we're characterizing, we want to get it all the way to this level of what gene is encoded and what happens when we mutate that gene at a molecular level, what is that doing to the protein function. Okay, so just to kind of see if we kind of have understanding of these different traits. One human disease that I think we've talked a little bit about cystic fibrosis, but this is caused by loss of function mutation in this gene called CFTR, which I think that stands for some conductance. I actually forget what the whole name of the gene, but it's a receptor that gets mutated. When it's mutated, it does not regulate chloride ion transport across the membrane. Because of that, you get this imbalanced osmotic. You have imbalanced osmosis, I guess, which leads to mucus forming along your epithelial linings. So all the places of the body that have epithelial cells get a buildup of mucus. So in cystic fibrosis, as you can see, one of the major symptoms is that you tend to have a lot of issues with your lungs. So that is an epithelial lining. If you get mucus in your lungs, what's going to happen in your lungs?
Your lungs will expand and surfactants and able to break that surface tension.
So that's probably an A&P explanation. Yes, definitely that probably affects that. But in addition, you see this cloudiness in the lungs. So what can that indicate? Infection, right? So if you have mucus, mucus is used to trap bacteria. But if you can't clear the mucus and get rid of it, we have infection. Your explanation is probably well beyond what I know about the lung function. But so they tend to, with cystic fibrosis patients, they usually have a hard time breathing, right? How many of you have seen the movie 5 Feet Apart or read the book? Nobody's seen 5 Feet Apart?
I know Cole Sprouse.
You what?
I know the actor.
Oh, yes, you should watch it. It's a, I think
I
it's
wish.
good. It's about two people who have cystic fibrosis, but they fall in love, but they can't get near each other because they will kill each other because they'll give each other an infection. And one of them gets, well, I won't kill the plot for you. So I recommend you watch it, but because you actually learn a lot about cystic fibrosis by watching it. With a lot of cystic fibrosis patients, they try to treat the symptoms, but there's no cure. And so usually cystic fibrosis patients will die in their like 20s or 30s because they end up with infections that they can't fight, and they end up usually succumbing to that infection. Yeah.
Would a lung transplant be curative, or is that?
Yeah. So in the movie, the girl is on a wait list for lungs, and she gets the lungs, and so. Anyway, yeah, so the problem with this is that we don't have a way to like treat the molecular basis of this. For this mutation, do you think it's recessive or dominant based on the sort of explanation that we have? Yeah.
Recessive.
Everybody think recessive? Yes, so loss of function. So if you had one copy of normal, or I shouldn't say normal, but functioning CFTR, that is enough to give you a wild type phenotype. So parents who have in their family history, if they have anybody in their family history who has cystic fibrosis, this is one common one that people will test for. They'll do genetic testing because it is so bad. I mean, essentially it kind of means that your life is going to be spent trying to avoid infections. And not only that, but there's other epithelial linings that are also affected. Your digestive system, your reproductive system. So it changes the way you live your life if you have cystic fibrosis. Okay, here's another trait that I'm sure you've heard of. I don't know how many of you have ever met somebody who had an extra digit. Maybe you haven't. How many people have seen Princess Bride? I feel like now that's getting like, I'm the only one who's, have people seen Princess Bride?
I never
What's the
saw it
Princess
as a
Bride?
child.
I know Princess Diaries, the one with Anne Hathaway.
Princess Bride. Okay,
Princess
put that
Bride
on
is
your
one of the
list
best
too. You have
movies
2
ever,
movies to watch
and you should
for
all
me
watch
so
it.
far. 5
My
Feet
name
Apart,
isn't
Princess
even
Bride.
my
Princess Bride is a classic. I really feel old because you guys haven't seen it. But Princess Bride,
1987.
what is it?
It
Is
came
it
out
from
in
1987? Oh, I would never know that one.
Oh, I recommend it. It's a really good movie. Anyway, in that movie, he's searching for this man with 6 fingers who killed his father. So anyway, that's the and then he says, my name is Indigo Montoya. You killed my father, prepare to die. But he's looking for this guy the whole time who has got 6 fingers. But that condition of having extra digits that can occur in your fingers or your toes, it can actually have a wide variety of phenotypes. So you can have as much as just an extra, you can have as much as a fully formed digit, like here, that person has 6 toes. In this case, you can see there are, what is this, like 7, and also like extra forming of the extension part. But it can be as minor as like a skin tag. So there's a variety of how severe polydactyly is, but that's the condition that's called polydactyly. It is caused by mutations in developmental patterning genes that are overactive, obviously, so they're creating more digits than they should. So based on that explanation, do we think it's recessive or dominant? Overactive.
Dominant.
Yeah, this is a dominant gene. So that usually people kind of associate dominance with common, right? You think, all right, if it's overriding, we should see a lot of people with polydactyly. But this I want to make sure in your minds that when you think of a dominant trait, you don't think it's common. How common a trait is has to do with how many people in a population have that allele. But there's actually quite a few dominant traits that are not that common. Yeah.
So what's it called when they have less fingers?
Fewer? Yeah, I think that could be caused by multiple different, like along the developmental pathway, there's multiple places where that can happen.
In high school, I think that as well.
Yeah. And I don't know, I know there's like a medical term to describe having fear, I don't know it though. Yeah. So this is an example of dominant trait. I have a little chart here of some others, just kind of for interest's sake. Albinism is a lack of pigmentation. That would be recessive because it kind of makes sense if you don't have pigmentation, you'd have to have 2 copies that are missing. Color blindness, that one's an interesting one. I actually just found out a couple of years ago that one of my boys is colorblind. I had no idea. He has really bad eyesight, and so we were at the doctor, and she was doing the color test. Have you guys all seen the colorblind test? And she was asking him to identify the numbers, and he was like, I don't see anything. And I had taught genetics for so long, I was like, oh my gosh, he's colorblind. I didn't know that. And that trait is actually an X-linked trait, which means that it turns out that I carry this. I'm not colorblind, and I have one boy who is colorblind, and my other child is not colorblind. So that means, I pass one on to one and one on to the other. For traits that are X-linked, who do you think gets them more? Is it males or females?
Yeah.
Males, yes, so it's, why is that? Why do males get X-linked more? Yeah, now.
Well, they only have one X chromosome.
Yeah, so they get what they get, right? And there's no way to like, you don't have a backup copy. So color blindness and muscular dystrophy, we talked about that a little bit when we talked about splicing. Both of those are X-linked, more common in males. Sickle cell anemia we've talked about as the blood disease, and then Tay-Sachs is actually a lethal. It's a disease that causes death in children, so it's another one that they will often test for if you're getting genetic testing done. Dominant traits, achondroplasia is dwarfism. That's dominant, so that's another example of something that is dominant but not that common. Huntington disease. Some of these other ones are just for traits like widow's peak. PTC tasting. I actually have some PTC strips in the back. If you want to see if you can taste PTC, I can get those out for the second half of class. But, so the takeaway here, dominant alleles are the most common alleles in the population. What is the answer?
False.
False. They are not, and there's two examples of why that is a false statement. That has to do with population genetics. You can also have in different populations, different traits are more or less common, right? If you think about red hair, for example, which ethnic group do you kind of associate often with red hair? Irish, right? Irish or European, yeah. Oh, I didn't know this. I mean, I didn't put two and two together. So maybe you have some Irish in your background. I don't know. Okay. So let's see. I guess it's 12:38. Let's do just a couple more slides and then we'll take a break. What I was thinking is we could do the scoring for the for our worms, and then that'll kind of break up the class, and then we'll come back to a little bit of genetics at the end. So we'll kind of let's do a couple more slides and then we'll do the lab part. Okay, so this is related to the idea of dominance and recessiveness, but what is the biological reason why we call it consanguineous matings, which is incest, basically. But why is it harmful from the perspective of genetics?
Because all of the recessive traits that aren't necessarily desired get mixed in to this, they keep getting mixed in the same family, which keeps popping out weird little babies like the Habsburgs.
Yeah, oh yes, so you have the reference here, yes. So this is the Spanish royal family, and as you know, royal families actually are highly inbred. I mean, if you look at, because they wanted, at the time, they wanted to keep it all in the family, so they set up all these crazy matings. This is just showing the family tree of Charles II of Spain. All of the blue ones are inbred mating, so either cousins or first cousins or like uncle and niece. This is just, it's kind of gross for us to think about that, but it's very common if you look at that. Another place we see a lot of inbreedings is with when we do pure breeding of dogs. If you look at, a lot of times the family tree for a dog, there is like, they have a lot of that mixing, and that, as you know, a lot of purebred dogs have health issues that are kind of augmented by that mixing. So the idea is that with your recessive traits, they're kind of increased if you have incest. In this case, for Charles II, he was kind of at the bottom of that family tree. I don't know if you saw him at the very bottom there. Charles II was kind of at the bottom of many inbred levels, but they measured his alleles. He had identical alleles for 25% of his genes, and for most people it's 0.25%. So that kind of shows you how much he had, a lot of recessive traits. He ended up suffering from several different recessive disorders as a result. So pituitary hormone deficiency and renal tubular acidosis were two that he had. All right. Okay, I think we'll take a break here, and then, so let's take like a 10-minute break, and then you can do your scoring for the worms, and then after that we'll do a little bit more of the genetics. Are you recording? I forgot that I never record.